It is genuinely wonderful to be here. Anyone who knows me knows that I have a profound caffeine dependency, so I appreciate this talk is between you and coffee, and know that I want to get it through as much as you do. So with that, we will get started. I want to start by thanking those folks who have invested in us. Juvenescence was our very first investors. They've been with us the entire way. Enormously grateful for their support. I've also met a number of shareholders on this trip, so deeply appreciated for other folks who have supported. And Sergey Young at the Longevity Vision Fund has also been enormously supportive of this work. This is, of course, an image of Prometheus, the god that stole fire and gave it to man, and his punishment from Zeus was to be chained to a rock. An eagle would come and eat his liver each day. It would regenerate over the night and only to be tortured by having the eagle eat it once again. And I raise this because the Greeks understood a fundamental truth that we're now on the bridge of exploiting clinically, and that is it is possible to enable in vivo organ regeneration. And that is precisely our mission at LyGenesis. Just a little bit about my colleagues. So Eric Lagasse is our Chief Science Officer. This story I'm about to tell you represents more now than a decade's worth of his work, sponsored by the National Institutes of Health, as well as the third pillar of our stool here is my Chief Medical Officer, Dr. Paulo Fontes, transplant surgeon, trained with Tom Starzl, the father of liver transplantation at the University of Pittsburgh, and very attuned to the unmet clinical need, which we'll be talking much more about, which is the demand for organs far outstrips supply. So it was often Paulo's job to tell families that their loved one would never get an organ because they wouldn't even qualify for the transplant list. So this is a very personal mission for him as well. Just a little bit, for those of you who saw my talk yesterday, this is a little less science, a little more business focused. So in terms of the key investment highlights as you think about LyGenesis as a clinical stage cell therapy company. So we are an allogeneic cell therapy company. We have cells that can regenerate a number of different ectopic life-saving organs. I'm going to walk you through our lead program, which is for patients with end-stage liver disease. We're on the cusp now. We just had our site initiation visit and are beginning to gear up for enrollment in a phase 2A trial at the Massachusetts General Hospital at Harvard, where we will be, for the first time, growing ectopic livers of invested U.S. capital. We'll talk more about the commercial proposition here, but unlike CAR-T therapies, CRISPR-based therapies, we have a remarkably low COGS. We almost have software-like margins for a cell therapy company, and we will talk more about that as well. So let me just kind of set the context here. If you need an organ today, there are some fundamental issues at work in that marketplace. One donated organ treats only one patient, so one-to-one correspondence. It also is the case that if you need an organ transplant, it is a major operative procedure. At least 50% of the patients who need one will simply medically not qualify for one. And just to give liver transplant as one example, the acute hospital cost for a liver transplant in the US is about $800,000. Using our technology, one donated organ can treat up to 75 patients. We are fundamentally changing the supply and demand calculus when it becomes to organ availability. I genuinely believe within the next decade, and certainly within our lifetime, we'll see an end of all organ transplant wait lists. They will really be relegated to the dustbin of medical history, along with mercury tinctures and leeches, although as I noted, leeches are making a comeback in some circles. It is also the case, using our cell therapy, we engraft ourselves using an endoscopic ultrasound. So this is an outpatient procedure under light sedation. It is a procedure in the US that's billed at about $2,000. So again, very cost efficient. It also takes only about 10 minutes to complete. If you're not familiar with endoscopic ultrasound, so the endoscope goes down the patient's throat in through the gut. And as it goes through the gut, there's a scope that as it transverses the gut wall, it can look for lymph nodes. And when it finds a lymph node, we take a 25 gauge needle down that endoscope, inject our cell therapy into these periduodenal lymph nodes that serves as bioreactors to regrow these functioning ectopic organs. Just a little bit about our pipeline. We have published in Nature Biotechnology and elsewhere the ability of the lymph node to serve as a highly effective bioreactor for a number of different tissue types. I'm going to be spending most of my time today talking about end-stage liver disease. There are actually multiple other liver indications. We've also shown the ability to grow an ectopic thymus, kidney, and pancreas, with, of course, some dramatic unmet medical needs for those various tissue types, which I will not belabor. There's a wonderful Arab proverb that I read recently that I just loved. It said, man fears time, but time fears the pyramids. I just love that quote in general, but it strikes me it is also the case that perhaps time should fear the lymphatic system. If you're not familiar, the lymphatic system is a remarkable product of our evolutionary tract and our ability to fight infection by educating and bioreacting T cells. So naturally what our lymph nodes do, right? When you have an infection where there are 400 or 500 of these bioreactors spread throughout our bodies, they really do something quite remarkable. And you heard a wonderful talk this morning on innate immunity. So these help with our T cells to fight various types of infections. We're leveraging that just the way that cancer does, right? Cancer probes the environment for ways to metastasize and spread. And what happens when cancer finds the lymph node? It finds a very well-vascularized space to grow tumors. Well, we're leveraging that same biology, but instead of growing a malignancy, we're growing therapeutic tissue. So what does this look like? So taking just an example of end-stage liver disease. So when you have a fibrotic liver, we take hepatocytes that are from donated organs. These are allogeneic. We have a drug master file now with the FDA. So it's a 70-step process, but we basically take a donated liver, and in about 6 hours, it takes 2 people in a GMP facility, just 6 hours, to have a solution ready to engraft our cell therapy into the lymph node. As I'd mentioned, we use the endoscopic ultrasound and are engrafting into the lymph node. Over time, what happens is the lymph node will actually disappear. So the lymph node is an initial bioreactor that starts this process of organogenesis, the development of a new organ within that lymph node. So over time, though, that lymph node will actually disappear, and what you're left with is a well-vascularized mini organ. In terms of what this might look like, and actually what it is looking like clinically, I should say now, when you have an organ donor, the extent to which a match is made, you always want that to go ahead and proceed because, again, of that supply and demand problem. But as was, I think, alluded to earlier in terms of throwing away the number of kidneys, just to give you the liver as another example, in the US, about 30 to 40% of all donated livers are discarded. And it's actually a problem that's getting worse in the US. The NHS here at the UK does a better job, and it's obviously more geographically tractable here in the UK than in the US. You only discard about 15% of the organs, but still, just as we're leveraging the lymph node as this natural, powerful bioreactor, it's also the case that part of our low cost of goods comes from the fact that we're using these donated organs that are not otherwise transplantable as the cell source. And this just goes to the fact there's a variety of in some ways U.S.-specific reasons for the increase in liver discard, driven largely by increasing rates of obesity and fatty liver disease, as well as an incredible tragedy of opioid overdose deaths in the U.S. 100,000 Americans died last year of overdose, and a lot of those overdose, when they're found, they've been down for many, many hours, and the liver enzymes will go through the roof, and transplant surgeons just are very uncomfortable using a lot of those organs. Just again, just to get a little bit into what I mentioned earlier in terms of the low cost of goods of therapies, because for all the excitement around cell therapies, if you've been following the CAR T space, and I think it's going to happen with CRISPR therapies as well, when you look at the attendant cost of producing those therapies, they are profound, right? And part of the commercial problem that some of the CAR T therapies are having are not with their efficacy profile. They are in many ways outstanding, but they are extraordinarily expensive on a per patient basis. Here, I'm comparing the cost of our program versus an orthotopic liver transplant, just to show you that, again, this is coming in at a fraction of the cost of a full organ transplant, so much so that the actual cost of the therapy then, even if you were to arbitrarily set it, this is just an example, by the way, you still have tremendous pricing headroom. Even if you set it at a per patient license of $200,000 a patient, you're still coming at a fraction. So from a payor perspective, right, it still is a very compelling return on investment that way. The last thing I'll just say is it is also eminently commercially scalable because you can have regional cell transplant facilities. Our work can really slot into any existing GMP cell processing facility. So the extent to which the University of Pennsylvania invested, just as one example, hundreds of millions of dollars in a state-of-the-art therapy for CAR T cell development. When we talked to big pharma and they ask me my infrastructure needs, I'm like, I need a 10-foot bench and a hood. So we can slot into any of those existing GMP facilities. A single facility then can provide the cell therapy for multiple hospitals. Each hospital then can treat multiple patients, which also helps on the scalability, just the commercial reach. So I've been involved in some drug companies where you have to mount huge sales forces, right, to try to address 30,000 GPs across the US. Here, we're talking 24 academic cell transplant facilities can service 90% of the US market. So eminently tractable just from a commercial perspective. All right, enough on the business. I'm going to get back to some of the fundamental science and how we know this works. And so I'm going to start talking about our lead indication, which is for end-stage liver disease. We're starting a phase 2A trial, as I had mentioned. The preclinical program here consisted of really a decade's worth of work by Eric Lagasse and Paulo Fontes. Started in FA animals. These are animals that have a genetically induced tyrosinemia type 1. It is a fatal liver disease. You keep these animals alive and relatively healthy by giving a drug that corrects that metabolic deficiency called NTBC. And as long as the animals have NTBC, they're fine. When you remove the NTBC, they die of a fatal liver disease in just a matter of days or weeks. It makes it a very compelling preclinical model because if you rescue these animals, something is happening, right? Because it's 100% death in this model. This is from Eric's Nature Biotechnology paper. This just goes, so the top pictures there on the left are the control animals that just received the buffer injection into the various lymph nodes. And then on the bottom row are those that have been injected with hepatocytes and have grown fully functional ectopic organs. So these are livers, behind the knee of an animal that should not be there, right? In terms of the staining and histology, I'll just note that broadly, these replicate the normal histology of the native liver. And in fact, in many ways, are indistinguishable insofar as they form the portal triads and the hepatic lobules that are characteristic of the native liver. These are just some of the survival data going to show that, again, when you get the successful engraftment, this is functional tissue that rescues the animals from otherwise fatal liver disease. We've, of course, cured many disease in mice. And so the issue is really, does it translate to large animals? And eventually, does it translate to humans? And so we've spent many years preparing the path for our clinical trial. This is the first of 2 different studies in pigs. This was done at the Mayo Clinic independently. These were FA knockout pigs. Again, here we radiolabeled the hepatocytes so we could actually watch the ectopic organs form. And again, we saw the same formation of central veins, bile ducts, liver lobules. So again, a replication of the normal histology of the native liver that again rescued these FA pigs. We then replicated these data again in pigs using a different model called the porticable shunt, which I won't belabor, but it's a surgical model of liver injury, and then met with the FDA and did a final study in dogs whose vasculature of their liver is a complete parallel to the human, so they're a very compelling case that way. Again, showing in that porticable model, able to rescue the dogs from that liver injury. This just speaks again to the functional tissues. This is rewinding a bit. My apologies to the previous pig study. These are the liver parameters. So if you look at blue, these are the control animals with no disease. Red are the FA animals who have had NTBC withdrawn, so they have a dramatic elevation in their liver biomarkers. And then the green represent the animals who have had the infusion of our cell therapy into their lymph nodes and grown these ectopic organs, showing a correction of the metabolic deficits. We've had several very productive meetings with the FDA. They've been enormously helpful and supportive. We are regulated through CBER Biologic and through the Office of Tissues and Advanced Therapies there. They helped us give input on the clinical trial designs. This is a dose escalating study. It's kind of a 4+4 design if you're familiar with clinical trial design. So an initial cohort of 4 patients look at acute safety and tolerability at 3 months. If it looks good from the Data Safety Monitoring Board, it'll then dose escalate to our low dose, which is 150 million hepatocytes. We inject 50 million hepatocytes per lymph node. So that's 3 injections. And then if all goes well, we'll dose escalate to a high dose, 250 million hepatocytes spread across 5 different of these periduodenal lymph nodes. These are patients with end-stage liver disease who are otherwise ineligible for transplant. We've also tried to be selective insofar as they have end-stage liver disease, but their life expectancy is years, not months. Right? And so this is an important limitation of this therapy. This will never be a therapy for acute liver failure, right? Because at the end of the day, the cells need time to recognize their new environment, to engraft, proliferate, and vascularize, form the liver lobules. Of course, the liver lobules are allogeneic. All of the vasculature that supports it is autologous, is native to the patient, right? Which could help to reduce the immune suppression burden. This first generation therapy will require immune suppression. We have several therapies in the works, next generation therapies that will hopefully be able to reduce or eliminate immune suppression. But kind of the low hanging fruit, the bird in hand, if you will, is this allogeneic therapy, which will require immune suppression. I love this quote too. If necessity is the mother of invention, discontent is the father of progress. While we're very excited about the progress we're making in end-stage liver disease, our chief medical officer was also very aware of another acute unmet need in inborn errors of metabolism. These are infants born with catastrophic inborn errors of metabolism where they have a normal liver, but they're missing a key enzyme that allows them to digest certain enzymes. Phenylketonuria is very well known. We announced just yesterday for the first time that we have launched a new drug development program because of some successful data we've generated to date. And it's going to first target maple syrup urine disease. Maple syrup urine disease is regarded as one of the most serious and significant inborn errors of metabolism. In general, these infants, if not on a carefully controlled diet, will develop a variety of CNS complications and die within a year. The only standard treatment for these children today is a lifetime of extraordinarily acute dietary restriction and or orthotopic liver transplantation. This new technology combines the hepatocytes that we've been using with a hydrogel with growth factors, but instead of going into the lymph node, and one of the expertises of LyGenesis, I should say, is this sort of intersection we have of deep anatomical expertise and deep cellular regenerative medicine expertise. And so this is combining a hydrogel with growth factors, and we're actually going to be using the liver. Oh, the video is not playing. This is a video. If it's possible for someone to play it, that's great. If not, that's okay. This is a novel catheter that we've developed that will go under the liver capsule. So the liver capsule can hold an enormous amount of fluid. And so we're going to use that liver capsule, go under that liver capsule with the catheter, inject this hydrogel that will allow us to engraft more than 100 times the current amount of cells that we can put in. And that's what these infants need. They need a therapeutic mass right away. They don't have a diseased liver with the pro-growth factors to trigger those hepatocytes to grow. And so we're going to create this artificial environment for them. So we're excited about this program as well. Lastly, just a little bit about our pipeline. So I've been focused to date exclusively on the liver. I'm just going to mention our work on the thymus, kidney, and pancreas. You heard about the thymus this morning. We have some more publications coming out, some very exciting data on the thymus. In general, we have already shown that we can take a young mouse thymus and graft it into an older mouse, and that is an actively functioning thymus within the lymph node that's actively educating T cells. And we're documenting the functional utility of regenerating the thymus. You saw a great picture earlier today from one of the talks on the involution of the thymus, right? So by late middle age, it is really no longer a very effective organ. And so the ability to potentially reboot that has potentially very exciting anti-aging implications. So a lot more to come on that front. A little bit in our work on the kidney. We published 2 papers on this just last year. The nephrons, so kidney tissue thrives within the lymph node. It goes very well, forms nephrons in these functioning ectopic kidneys. Here we have what I will just summarize as a plumbing problem, because at the end of the day, you do have to do something with the urea. And so we have more work to do to see whether engraftments of lymph nodes adjacent to the bladder will help fistulas to form to drain the urethra from these ectopic kidneys. So more to come on that front. And we're very actively and have some very exciting research, ongoing research collaborations that we'll be announcing in the near future. We have also shown that if you engraft pancreatic islets into lymph nodes, they will glucoregulate and correct they will insulin sensitize and glucoregulate otherwise diabetic animals. And so very excited about this program as a potential treatment for type 1 diabetes as well. And we will have more to come on the diabetes program as we move forward as well. Lastly, just in summary, so we are very excited to kind of represent a translational clinical stage cell therapy company in the regenerative medicine space. We're very excited about our core indication in end-stage liver disease, a lot of potential follow-on indications as we've mentioned. And we've also, of course, as any young biotech does, right, have been aggressively pursuing intellectual property. We own both issued and pending intellectual property on the use of the lymph node as a site for organogenesis, alternative sites for that, as well as a variety of around the use of endoscopic ultrasound to engraft our cells into the lymph node. So with that, thank you very much for your time and attention.